Good morning, class. Once again, we continue in the mitosis division. We already discussed the general cell structure and all the structure which involves in the cell division. Now we start with the prophase. So prophase, I already told you there is involvement of the some structure in the cell, and what changes occur during the prophase. The first, the nuclear membrane and nucleolus, they both start disappearing. Clear? Then the centrosome, if it is animal cell, one thing is clear, if it is animal cell, then centrosome is split and produce the copy, and that is called centriole. And that centriole start moving towards the opposite pole. It means one in inside this side and another in opposite side. So these changes occur outside the nucleus. It means the nuclear membrane start disappearing, nucleolus also start disappearing, centrosome is split and produce two centrioles. The centrioles start moving towards the opposite pole. You can see in the next diagram. The chromatin network which present inside the nucleus, that chromatin network start condensing. They start shortening. They start thickening. And due to that thickening, shortening, condensing, they are visible like the thread, and further they are visible like the chromosomes. They start appearing as a chromosomes. And when we see this chromosome, so they are having the two chromatids. Means you can see in this diagram there is two chromatids, and both chromatids are connected with each other by the centromere, a structure where the chromosomes are connected. I just try to draw here one more diagram only for the chromosome. So this is chromatids, and they are considered as sister chromatids, and both chromatids are connected at a point, and that point is called centromere. Clear? So the genetic material, they are become short, thick, and they visible as a chromosomes having the two sister chromatids, and they are connected with the centromere. And when we see the final stage of the prophase, so what happened? The centrioles reach to the opposite poles. So centrioles reach to the opposite poles. Around the centrioles there is rays-like structure. They are called astral relays. Nuclear membrane, nucleolus totally disappear, and in between the centrioles there is formation of the spindle fiber. The fibers appear, proteinaceous fibers appear in between the centrioles. That is what? Spindle fiber. And chromatin network become as a chromosomes, and each chromosomes having the two sister chromatids. Both sister chromatids are connected with each other by the centromere. So this is the last or final stage of the prophase. So here we can say this is early prophase, this is middle prophase, and this is late prophase. During the examination, question can asked on any diagram, and you are going to write only and only one thing, that is what? Prophase. So all these diagrams representing the prophase, because there is no early, middle and late in the syllabus of the 10th class. Clear? So once again I repeat, what happened during the prophase? The nuclear membrane, nucleolus disappear in this stage. The chromatin network gets separated and become as a chromosomes. Each chromosomes contain the two sister chromatids. Both sister chromatids connected with each other by the centromere. Centrosome, if it is animal cell, then centrosome is split and produce two centrioles. These centrioles move to the opposite poles, and in between the centrioles there is a fiber-like structure that is called spindle fiber. So this is the end of the prophase stage, and now cell enter into the metaphase. Clear? Now come to the next. After completion of prophase, there is next phase that is called metaphase. What happened in the metaphase? I already discussed. This is the prophase. So all the chromosomes, you can see they are scattered inside the cell membrane, and these scattered chromosomes start moving towards the equatorial plate. Equatorial plate means in the center. Clear? And all the chromosome, they are attached with the spindle fiber. You can see in the diagram, they are what? Attached with their spindle fibers. They are what? They are spindle fiber, which is present between the centriole. So all the chromosomes arranged at a equatorial plate, it means in the middle region, and chromosomes attached with the spindle fiber by their centromere. By what? By their centromere. And this is the specific condition of the mitosis cell division, and in this phase we can understand by a single diagram. So I draw only a single diagram. So here this is prophase. In this you can see the chromosomes are not arranged at equatorial plate, but in this metaphase stage all the chromosomes are present, which is present in the cell. They are arranged at equatorial plate. In this diagram you can see there is total number of the chromosome 4 chromosomes. So there is total 2 pair of chromosomes present in the cell, and they are arranged at equatorial plate. One more thing, this centromere is also considered as kinetochore. Clear? So this is about the metaphase stage. Okay. Next phase, that is anaphase. So I already told you during the metaphase all the chromosomes are arranged at equatorial plate. Now what happened? The chromosomes which is present on the equatorial plate, due to the contraction of the spindle fiber, this chromosomes split from the centromere. Just I draw one chromosomes here, and then I try to explain. This is what? This is centromere, and these are the sister chromatids. So what happened? This chromosome is split from this centromere, and they start, the sister chromatids start moving towards the opposite pole. So this is what? Splitting of the chromosome. From where? From centromere. Clear? So all the chromosomes which is arranged on the equatorial plate, due to the contraction of a spindle fiber, the attached chromosomes split and sister chromatids start moving towards the opposite pole. And slowly these chromosomes reach towards the opposite poles. So this is very important stage of the mitotic phase or karyokinesis, because here the chromosomes split and equal number of chromosomes reach on opposite poles. Clear? There is no change in the number of chromosomes. Here it's a 4, so 4 chromosomes reach on this pole and 4 chromosomes reach on the another pole. So there is no change in the number of chromosomes. This is the unique feature of the anaphase of the mitosis division. Clear? Now come to the last phase of the karyokinesis, that is what? Telophase. So after the completion of anaphase, the cell enter into the telophase. What happened in this? A furrow is start developing in the cell membrane, and the chromosomes which are rich on the opposite poles, they start condensing and converting as a chromatin network. Okay, so what happened? They start converting as a chromatin network. There is conversion of the chromatids into the chromatin network. Next, the spindle fiber which present between this, they start disappearing, they disappear, and the nuclear membrane and the nucleolus start appearing. So what thing start appearing? Nuclear membrane and nucleolus. So this is what? This is nuclear membrane and this one, it's what? Nucleolus. So what thing? What is the specialty of telophase? The chromatids, they start converting into the chromatin networks. Clear? Then the nuclear membrane start reappearing, nucleolus start reappearing, the furrow start developing in the cell membrane in animal cell. So this can be considered as what? The cell furrow. And finally, by the completion of the telophase, the two nucleus is formed inside the cell. The well developed nuclear membrane, nucleolus, chromatin network, nucleolus, chromatin network, nuclear membrane, and centrioles duplicated to form or convert into centrosome. Clear? So by this way, the two nuclei are formed inside the cell, and both nuclei contain the same number of the chromosomes which is present in the mother cell, which divide by the mitosis division. So during the karyokinesis of the mitosis division, there is no change in the number of chromosome, there is no change in the set of the chromosome. The equal number of chromosomes which is present in the mother cell is also present in the daughter nuclei. So here the karyokinesis of the mitosis completed, and now cell enter into the cytokinesis. Clear? If suppose it is plant cell, then what changes we observed? So if it is plant cell, then one thing what we draw in each diagram, that is what? Centrioles. This centrioles is not in the diagram. So by this way you can identify or recognize if there is no centrioles, it means the diagram represent the plant cell. And another thing, if one more covering is present, I already discussed this thing in the beginning. If it is plant cell, then what thing is not present? So if we see the changes, what thing? That is what? There is no centrioles, there is no astral rays in the plant cell, and one more covering, that is cell wall, is present outside the cell membrane. And one more thing, there is no formation of furrow in the cell membrane, because cell wall which present around it, that is what? That is the rigid covering. So this is the basic difference of the karyokinesis in plant cell and animal cell. In the plant cell, there is no centrioles, there is one more protective covering, that is cell wall. Normally question arises, the given diagram is plant cell or animal cell, justify your statement on the basis of the support. So to support, centrioles absent in the plant cell, plus there is presence of the cell wall. Clear? In each diagram If you remove the centrioles and one more protective covering you drop, then that is plant cell. And all other things which present inside the cell membrane, they are what? Same in the plant cell and animal cell. So by this way, the karyokinesis in the plant cell and animal cell during the mitotic division is completed, and we discuss the cytokinesis. Cytokinesis is different in the plant cell and animal cell. Clear?